Hi, everyone. My name is Upendra Marathi. I'm one of the co-founders and serve as the CEO of Seven Hills Pharma. So I'm pleased to announce Seven Hills will be a clinical stage pharmaceutical company in about 2 weeks. We're going to be dosing our very first cancer patient. And so we're developing these integrin targeted agonists to prime a de novo T cell response and addressing PD-1 refractory disease. So with the benefit of about 34 million from the state of Texas and the NCI, we've been able to start this brand new biological concept and now work all the way through the clinic. So I'm going to give you a story about Mr. Magoo and Baby Bugs Bunny. Okay. So if you think about Mr. Magoo as someone exemplary of melanoma and non-small cell lung cancer, and it might end up going on a PD-1 inhibitor, and we know vast majority of the people will progress on a PD-1 inhibitor. And so we believe we can actually take those progressors and take an integrin targeted agonist called alantegamod and combine it with ipilimumab and drive de novo T cell activation and improve clinical outcomes. So I'm very pleased to announce that we now have a new compound to address heme malignancies in using a small molecule integrin targeted agent to deliver more stem cells to the bone marrow niche and deliver the promise of cord blood transplant as a curative regimen for the treatment of AML and ALL. So here's the biology. This is instructive for our vaccine program and the checkpoint inhibitor program. So in order to mount an antigen-specific immune response, obviously you got to recruit T cells to the sites of a given tumor or activated dendritic cells. So this recruitment requires taking patrolling cells to engage the endothelium that expresses VCAM and ICAM. And this is a rate-limiting step. If you don't get this and get intravasation, you won't get any priming. And this is fundamentally the basis of Humira as an anti-inflammatory agent. So these integrins are also required not only for recruitment, but also as co-stimulatory molecules in training a new T cell. So engagement of an antigen-presenting cell to a given T cell through MHC requires a secondary signal, and these integrins can also serve as this. So what we have is an allosteric activator, and this alantegamod molecule is a dual activator of alpha-4 beta-1 and alpha-L beta-2 that dramatically increases the potency and adhesive potential here or here by about 100-fold. Okay, so these are not direct agonists, but they're positive allosteric receptor modulators. And obviously, if you can generate high-quality T cells, you can prime both a T cell response that's instructive for better T cell immunity as well as B cell immunity. And by the way, these compounds are synergistic with just about every single vaccine we've tried. So here's the overall focus of the company. On the solid tumor program, this compound alantegamod, we finished a healthy volunteer study to find the dose range, and with the help of CPRIT and the NIH, we'll be dosing our first cancer patient here, and we're funded through a dose escalation for the next 2 years in the company. So I'm particularly excited to introduce you about this 935 compound that can deliver double the homing of stem cells to the bone marrow niche, and we're just awarded about $5 million from the state of Texas to bring this compound to an IND level. And so we've been running the company almost exclusively on non-dilutive funds. Historically, we've got about 34 million, of which 25 million is active. So with a little bit of angel money and this non-dilutive money, we've been able to go from biological concept and hopefully all the way through clinical proof of concept. And so let's drill down on the solid tumor program a little bit. So if you're Mr. Magoo with PD-1 refractory melanoma, odds are you're going to go on dual checkpoint blockade, and that does work with about 20% response rate. Okay, and you generate T cells, antigen-specific T cells, but the price you pay is heavy toxicity. So we think we can actually use alantegamod as a substitute for nivolumab in generating new T cells. And if you do that, you can drive co-stimulation, improve tumor responses, and importantly, with no added toxicity. And we've tried to push the toxicities in inflammatory models until we're blue in the face. And it's highly selective for areas where we have very low levels of ECAM and ICAM. So in every one of those situations, you get a better antigen-specific immune response with no added tox. So here's some instructive data. This is a B16 BL6 melanoma mouse model that I believe is one of the few predictive models in IO. So it's a vaccine model that's totally refractory to, is this working? No. It's totally blue line. It's totally refractive to a PD-1 monotherapy. If you use a CTLA-4 inhibitor, you can generate new T cells, and you get about 20 to 30% long-term response with total immunity. And if you add 349 alantegamod, you can triple that complete response rate on the right panel. And if you look in the tumor microenvironment early on for antigen-specific T cells, there's a nice increase in antigen-specific T cells. So here's a study that we're rolling into here next week. It's a phase 1b study. We're doing monotherapy run-in to ipilimumab followed by nivolumab. And it's a fundamental deviation for trial designs in this particular space. Here we're trying to use an immunomodulator in combination with IPI, a full course of 3-month cycle of IPI to generate new T cells and then prevent exhaustion thereafterwards. So we're starting at the optimal pharmacokinetic dose, thank you FDA, at 100 milligrams, doing combination dose escalation, and then follow on with an expansion in melanoma in a placebo-controlled fashion at 2 different doses. Thank you, Project Optimus. So I'm going to switch gears here and introduce you to the leukemia program. So if you're a high-risk AML patient and ALL patient, first-line therapy is really to get you a cord blood transplant and get you a good match. Up to 70% of the people that are candidates for stem cell transplant can't get a match. And this is particularly true for non-white folks, Indians, Asians, Blacks, Hispanics, and so forth. So umbilical cord blood is a readily available stem cell source. It's ideal in every way. It's effective. It has lower risk for graft versus host. It requires a less restrictive match. But the dilemma with this thing is very slow to engraft. Okay, and I think with a single dose of a compound, I think we can address this issue. So here's the biology around this. So stem cells express alpha-4 beta-1 on their cells, and that is absolutely essential for the homing to the bone marrow niche. Okay, so particularly in cord blood-derived CD34 positive cells, this integrin is in inactive state. And because it's in an inactive state, not enough cells actually would get firm adhesion to the endothelium and migrate in. And so what we believe we can do is use 935, an integrin-targeted VLA4 selective agonist, and promote cell adhesion and deliver more T cells. And if you're able to do that, you won't need any CAR T-based therapies or so forth. And if you do that, you deliver more stem cells, and you can accelerate neutrophil recovery. And we're particularly attracted to this program because I think we can use absolute neutrophil counts as this pivotal surrogate marker for potential approval. So this is an acute homing model where we've taken CD34 positive human-derived cord blood cells into irradiated mice and look for homing into the bone marrow on the left side and look for finished blood products thereafterwards. What was striking was that contemporaneously with the cord blood infusion, if you give a single dose of this compound, you double the homing of the stem cells to the bone marrow niche. And they were remarkably instructive in repopulating, I think, the bone marrow. And so I'm going to skip over this. I'm fortunate to have a really remarkable group of people working with us. We're all investors in this, and this management team is funded about 90% of the equity capital that's gone into the company. So a lot of this work, it would not have been possible with our scientific advisors and founders. So Mary Laughlin and Apatsalia Tisaberidu, they're going to be running the phase 2 program. And by the way, we're a totally virtual company. So we've been able to go from concept to all the way through phase 1 for less than $13 million, including funding the company. And it's incredibly efficient. Typical cost is about $75 million. And we hope to leverage about $35 million in grant money, substantially with non-dilutive capital to continue to add value and drive liquidity. So we're pre-marketing a potential A round. I'd love to get thoughts from you all. Whether we should be out in this environment to be able to try and partner for where we are or even try capital, right? So we have the luxury of waiting for about 2 years for these clinical readouts before we ever go out. And if there's any angel investors in the room, we're in the process of closing out a convertible note. So I'll leave you with these following thoughts. This is clearly an opportunity to do well as do good. We've been exhaustively diligenced by the state of Texas in every freaking possible way. And based on this, we've gotten about $25 million in non-dilutive money and are about to add a substantial amount of value over the next 3 to 4 years. And so we've all been around the block with multiple approvals, and we've defined a brand new model on how to fund early stage innovation and show clinical proof of concept. And so I'd love to get any feedback for you on our business development strategy or our fundraising strategy. So thank you very much for your interest. I look forward to continuing the dialogue.